Hi, my name is Sabine,
Hi, my name is Amy.
and we completed Exercise 1, DNA Extraction of Biomolecular Techniques.
Overnight, we hydrated split peas in 200 milliliters of distilled water in a clean plastic cup.
That's what you can see us pouring out into a mortar here. Before we started doing this, we already got all of our safety gear on, including gloves, lab coats, and safety goggles. The goal of our experiment is to extract the DNA from the split peas. The first step of this process is the mechanical breakdown of the cells. Essentially, we will be using what's called a mortar and pestle to physically grind the peas to the cellular level to access their DNA.
It feels like a cooking
Okay. So now
Here, we use a large graduated cylinder to measure and add 100 milliliters of distilled water to an empty plastic cup. The reason we use distilled water is because it's free of contaminants, which ensures a more accurate result. When you measure with a graduated cylinder, use the bottom of the curve called the meniscus to measure the amount. We add water to assist in the mechanical breakdown of the peas to access their DNA.
It's a little too much.
1 over.
If you ever pour too much into a graduated cylinder like we did here, you can always pour out the excess, and in the case of water, into the sink. Graduated cylinders are actually more accurate at measuring volume than beakers are, so that's why we opted to use a graduated cylinder. After pouring out and ensuring that we had the correct amount of water, we will be putting it into the plastic cup that we prepared earlier.
As you can see, we are pouring the contents of 2 salt packets into the water that we will pour onto the peas. We use salt in DNA extraction because it helps neutralize the negative charges on DNA molecules, which allows them to clump together and precipitate out of solution, which separates the DNA from other cellular components, like proteins.
After pouring out all of the salt from the 2 packets, we need to stir the water to make sure that all of the salt combines with the water. We use this object called a stirring rod to stir the water and make sure that all of the salt is fully dissolved.
Now the salt and water mixture is ready for us to pour into the mortar and pestle containing the peas.
After fully preparing our water and salt mixture and pouring it into the mortar with the peas, we were ready to begin the crushing process of the peas. We do this by using what's called a pestle to apply force in a circular motion onto the peas to fully crush them and to create a paste-like consistency. After some time trying to crush the peas, we ran into a few issues while using our mortar and pestle. The first was that the peas would move around in the mortar, so it would be difficult to crush them. And the second was that with all of the water in the cup, it would easily either overflow out of the mortar or cause the peas to float around, making it even more difficult to crush them. We needed to find a way to solve these problems because otherwise it would have taken a really long time to crush the peas properly. Our first solution to this problem was to decant some of the liquid from the mortar out into a plastic cup that we could combine again later to form our paste-like consistency. This definitely helped our process. As we continued attempting to crush the peas, we continued to run into some problems. After pouring out some of the water, it was slightly easier to crush the peas because we didn't have to worry as much about water pouring out over the sides, but having less water allowed the peas to adhere to the walls of the container and sometimes fall out. So we quickly realized that we needed to find another solution to this problem.
Our next solution was to find a larger mortar and pestle and transfer the peas, which solved our problems because it gave more space for the movement.
DNA is the genetic material that is held within all cells. We complete this process of crushing the peas to mechanically or physically break down the membranes present within cells that hold the DNA. After completely mashing the peas and recombining the water that we previously decanted, we were left with the slurry-like consistency that we were aiming for, and this completed the preparation stage of our experiment.
Now we will use a large graduated cylinder to measure and add 20 milliliters of isopropyl alcohol to a clean plastic cup. We placed the cup containing the alcohol in a freezer in the lab.
We will be using this alcohol later on in our experiment.
We placed a clean plastic cup on the digital scale and tare the scale. This resets the display to 0 to account for the weight of the cup, allowing us to weigh the contents inside. Then, we add liquid detergent from the bottle to the cup until the scale reads 7.5 grams. We use a rubber band to affix the cheesecloth to the top of the cup containing the detergent.
This creates a mechanism that we can use to strain out the chunkier parts of the pea slurry that we created earlier to make sure that we only included the liquid.
Then, we carefully poured the crushed pea slurry onto the cheesecloth, allowing it to drain into the cup.
Combining the pea slurry with the detergent allows us to chemically break down the cells or lyse them in order to access the DNA held inside of them. We made sure to collect as much liquid from our pea slurry as we possibly could because that is what contains all of the DNA that we would be extracting.
Here, we remove the cheesecloth from the top of the cup and used a glass stirring rod to gently stir the mixture without producing froth or suds. We placed the plastic cup containing the pea and detergent mixture in a refrigerator in the lab for 10 minutes. Here, we used a large graduated cylinder to measure and add 20 milliliters of chilled pea and detergent mixture to a 25 by 150 millimeter glass test tube. We set the cup containing the remaining pea mixture aside. Then, we carefully transfer half the contents of the papain vial to the mixture in the test tube. Papain is a protease, or an enzyme that breaks down proteins found in the papaya plant.
Using a protease like papain removes all unwanted proteins and enzymes from your solution, ensuring the integrity of your DNA sample.
Once more, we use a clean glass stirring rod to gently stir the mixture. We retrieved the cold isopropyl alcohol and slowly poured it into the test tube, allowing the alcohol to flow down the inside walls of the test tube.
The alcohol is used to precipitate the DNA present within the pea mixture. DNA is soluble in water and typically insoluble in alcohol. When the cold alcohol comes in contact with the pea mixture, a film of DNA forms in between the pea mixture and the alcohol mixture, which sits on top of the pea mixture because it is less dense.
We held the test tube stationary for 1 minute while the reaction between the alcohol and DNA proceeded. Lastly, we examined the alcohol and interface layers of the solution in the test tube for DNA. The alcohol is the clear layer on top, while the interface layer is the white strip in the middle. We used a clean glass stirring rod to determine the consistency of the DNA.
Yes.
How would I notice? I have no idea.
So they used about 15 to 25 milliliters of pimental samples, and we brought it down to 5 milliliters to test to see if you needed a certain amount less than a certain amount they used before. And
What's in a requirement for contamination and toxicology?
It looks like...
Patrick?
How did this research opportunity help you grow as a leader?
so this allowed me to learn